Fusion of two dissimilar sexual units or gametes is termed as fertilization. In gymnosperms, pollen grains land directly on new cells, while in angiosperms, pollen grains fall on the stigma. In angiosperms, fertilization being completed as follows. Germination of pollen grains. After being deposited on the stigma, the pollen grain absorb the liquid from the moist surface of the stigma, expands in size, and can protrudes out through a germ pore. A small tubular structure is known as pollen tube is elongates, makes its way down the tissues of the stigma and style. Only distal part of the pollen tube possesses the living cytoplasm. Stigma plays an important role in the germination of the pollen tube. Stigma secretes fluid containing liquid sugars, gums, and resins. The chief function of the stigmatic secretion is to protect the pollen grains as well as stigma from desiccation. After arriving to the wall of the ovary, the pollen enters through the micropyle or some other route. Means in midst of the ovule or chalagal route or through the micropyle. Micropyle means porogamy, through the chalazogamy. In midst route, that is called mesogamy. In normal condition, pollen tube enters through the micropyle is known as porogamy. Pollen then enters through the chalaza is known as chalazogamy. This is discovered by the Trube in 1891 in Casuarina. The period between pollination and fertilization is different from plant to plant. Usually, this period varies from 12 to 48 hours. According to P. Maheshwari, 1849, the temperature is responsible for the growth of the pollen tube. Gammatic fusion. In normal cases, one male gamete fuses with the egg and form a zygote, and second travels a little farther and unites with the secondary nucleus. This process is known as double fertilization. In 1898, Navashin and Gaignard, 1899, as second male gamete fuses with the secondary nucleus, which is diploid in condition, a producing triploid sperm nucleus. This fusion is called a triple fusion. Result in formation of the endosperm. Thus, in an embryo sac, there are two fusions. One is syngamy between the male gamete and egg, triple fusion, and therefore this phenomena is called triple fusion, and therefore this phenomena is called double fertilization. Significance of the double fertilization. The embryo, during its growth and development, it receives the nourishment from the endosperm, which is a product of double fertilization. This process is also gives rise to the required energy to the polar nuclei, which fails to divide further. Since the endosperm nuclei are the resultant of double fertilization, they are characterized by the maternal and paternal chromosomes, and thus the endosperm represents the physiological aggressiveness due to hybrid vigor. Fusion are male and female gametes and as well as double fertilization is equally responsible for the production of the viable seeds, because absence of any one of these two causes lethal effect and viable seeds are not produced. Types of endosperm formation. There are three general types of endosperm formation: nuclear type, cellular type, and halobial type. Nuclear type. In this type, the first division and usually several divisions are unaccompanied by the wall formation. The nuclei either remain free or in later stages they may become separated by the walls. As divisions progress, they may become separated by the walls. As divisions progress, the nuclei are being pushed towards the periphery. Thus, large central vacuole is formed. Often the nuclei is specially aggregated at the micropolar or chalazial ends of the sac and form only a thin layer at the sides. Generally, the endosperm nuclei in chalazial part of the embryo sac have been observed to be larger than those in the micropolar end. The number of nuclear designs varies in different plants. Endosperm coccus nucleus of a palm deserves special mention. Here, the primary endosperm nucleus undergoes number of nuclear divisions. When the fruit is 50 mm long, nucleus are filled with watery fluid or milk-containing free nuclei and fine cytoplasmic particles. At a later stage, when the fruit is about 100 mm in length, the liquid shows in addition of free nuclei. Gradually, these cells and free nuclei set at free periphery of the cavity and the layers of the cellular endosperm form, and this become coconut meat. On maturity of the coconuts, the endosperm does not have nuclei or cells. In areca nut, the development of the endosperm is like that of coconut, but the embryo sac is small and it is completely filled up by the growth of the endosperm and later becomes very hard. The nuclear type endosperm formation is most common type found in wheat, rice, maize, sunflower. Cellular type. In this type, first and most of the following divisions followed by the wall formation. The first and most of the divisions are accompanied by wall formation, and thus the sac divided into several chambers, some of which may contain more than one nucleus. The first wall usually transverse but sometimes oblique and vertical, and in some other cases the plane of division is not constant. On the basis of orientation of the walls following the first two or three divisions, this type of endosperm have been further divided into subtypes. Halobial types. This type is frequently found in the members of the halobial, and in some other cases, the plane of division is not constant. On the basis of orientation of the walls following the first two or three divisions, halobial types. This type is frequently found in the members of the order halobiales. This type is intermediate between the cellular and nuclear endosperm. In this type, first division followed by the transfer wall, resulting in a micropolar end and chalazial end. Further divisions are free nuclear and may be formed by micropolar chamber only. Eremerus is an example of a typical halobial endosperm. Here, primary endosperm divides transversely forming two chambers, a large micropylar and a small chelagel chamber. Free nuclear divisions occur in both are more rapid in micropylar end or micropylar chamber. When chelagel chamber has 8 nuclei and micropylar chamber has 16 nuclei, and when there are 30 to 30 nuclei in the chelagel chamber, the micropylar chamber considerably a large number of nuclei. In older ovules, the chelagel chamber begins to degenerate. Finally, when formation takes place in the micropylar chamber, the chelagel chamber almost crushed, shows only a few disorganized nuclei. Development of embryo in dicots. According to Suiges, the mode of origin of four-celled pro-embryo and the contribution made by each of these cells makes the base for the classification of embryonal type. However, development of dicot embryo in Capsella bursa pastoris crucifer type for first time, Einstein in 1870 worked out the details of the development of embryo in Capsella bursa pastoris crucifer family, crucifer member. Wood spore divides transversely, resulting in two cells, a terminal cell and a basal cell. The cell towards the micropylar end of the embryo sac is suspensor cell, that is basal cell. The other cell makes the terminal cell, means embryo cell. The terminal cell subsequent divisions give rise to the embryo, while the basal cell contributes the formation of the suspensor. The terminal cell divides by vertical divisions, forming a four-celled shaped embryo. In certain plants, basal cell also forms hypocoti, the root end of the embryo. In addition of suspensor, the terminal cells of four-celled pro-embryo divides vertically at a right angle to the first vertical wall-forming four cell. Each of four cells divides transversely, forming actant stage. The four cell next to suspensor, termed as a hypobasal or posterior actants, while the remaining four cells make the epibasal or posterior actant. Epibasal actant give rise to plumule and cotyledons, whereas hypobasal actant give rise to the hypocotyle. Exception to its tip cell. Now all 8 cells of actant divides periclinally and form inner outer cells. The outer cells divides further by anticlinal divisions, forming peripheral layer of epidermal cells. The dermatogen. The inner cells divides longitudinally and transversely, forming peribelum. Beneath the dermatogen and the plerome in the center region, cells of peribelum give rise to the cortex, while that of plerome forms the stele. At the time of development of actant stage of embryo, the two basal cells divides transversely, forming a 6 to 10 celled filament. The suspensor, which attains the maximum development by the time the embryo attains globular stage. The suspensor pushes the embryo cells down into endosperm. The distal cell of the suspensor much larger than the other cells and act as a ostotrium. The lowermost cell of suspensor, termed as hypophysis, by further divisions. The hypophysis gives rise to embryonic root and root cap. With the continuous growth, the embryo becomes heart-shaped, which is made up of two primordia of cotyledons. Cotyledons appears on either side of hypocotyl. The mature embryo consists of a short axis and two cotyledons. Most of dicotyledons, the general course of embryogenesis as followed as seen in Capsella bursa pastoris. There is no essential difference between the monocotyledons and dicotyledons regarding early cell divisions of the proembryo, but the mature embryo is quite different in two groups. In what? Monocots and dicots. Here the embryogeny of Sagittarius has been given as one of the example. The zygote divide transversely, forming basal cell and terminal cell. The basal cell, which is larger, lies towards the micropylar end, does not divide again, but become transformed directly into large vascular cell. Terminal cell divides transversely from two cells. Of these, lower cells divides vertically, forming a pair of juxtaposed cells, and the middle cell divides transversely into two cells. In the next stage, two cells once again divide transversely, forming quadrants. The cell next to quadrant also divides vertically. The cell next to upper vascular divides transversely several times. Transversely, quadrants divides transversely again and forming octants. 8 cells being arranged in two tiers of 4 cells each. The result of periclinal division, dermatogen is formed. Later, periplum and pleurum are formed and differentiated. All these regions formed from the octants develop into a single terminal cotyledon afterwards. The lowermost cell of the three-celled suspensor divides vertically from plumule or stem tip. Cells of R form radicle. The upper 3 to 6 cells contribute to the formation of the suspensor.